Hello everyone, I'm Pierre Mariotte, and in this video I'm going to present you the findings of my study published in Journal of Ecology entitled Stoichiometric N2P Ratio Flexibility and Mycorrhizal Symbiosis Favor Plant Resistance Against Drought. Before I start, I would like to thank the editors of the journal who selected my article as editor choice of the issue and now as highly commended paper for the Harper Prize 2017. I performed this study during my postdoc in Australia. As you may know, Australian soils are very limited in nitrogen and phosphorus. Furthermore, drought events are forecasted to increase in Australia, even more so than other parts of the globe. Drought reduces CO2 assimilation, decreases the mobility of nitrogen and phosphorus, thus reducing plant uptakes. Co-limitation in nitrogen and phosphorus is an important factor influencing plant productivity. Then, measuring the N2P ratio in plant tissues can inform about nutrient limitation, but also how the plant adapt to perturbations. For example, a plant N2P ratio lower than 10 would reflect nitrogen limitation, while a plant N2P ratio above 20 would reflect phosphorus limitation. Plants can vary in their degree of homeostasis. For example, some species, such as in the bottom left graph in this figure, can better maintain constant N2P ratio independently of changes in the resource, here N2P ratio in the soil. These species would be considered more homeostatic than, for example, the species in top left graph in this figure, which tend to be more flexible, because leaf N2P ratio is more dependent on the soil N2P ratio. Previous studies showed that higher degree of homeostasis would lead to dominance of the species, as they are more able to withstand changes in nutrient resources. Furthermore, these species are expected to better resist perturbation, but it is also high energy demanding. These results came in contrast to what I previously observed during my PhD in Swiss grasslands. In a previous study, published in Journal of Ecology in 2013, I found that dominant species were the most impacted by drought, while subordinate species were able to maintain their biomass under drought and mediate drought effect on ecosystem functioning, likely due to specific association with fungi. When I arrived in Australia, I found the same pattern in grasslands, with the biomass of dominant species greatly reduced by drought, while subordinate species were not affected. I wanted to better understand by which mechanism subordinate species better resist water stress. Do subordinates possess better water use efficiency, or can maintain constant plant N2P ratio under drought? Another important factor in Australian grasslands is arbuscular mycorrhizal fungi, because of the soil limitation in both nitrogen and phosphorus. Plant colonization by mycorrhiza can increase under drought, improve water use efficiency, and influence plant N2P ratio. So I then wondered if subordinate species are more colonized by mycorrhiza than dominant species, and if it can improve the resistance to droughts. When I arrived in Australia in 2015, I had the chance to work with Alberto Canarini, a PhD student who already started a drought experiment under the supervision of Feiki Dijkstra. The experiment included 8 plots that received ambient precipitation and 8 plots that received 50% of precipitation, all organized in 4 blocks along a slope. I collected 2 plant species in each plot, the most dominant species, Paspalum dilatatum, and the subordinate species, Cynodon dactylon. Both are C4 plants. I harvested 3 individuals for each species and plots, including the shoots and roots. I then measured concentration in carbon, nitrogen, and phosphorus in leaves, and natural abundance isotope 13C as a proxy for water use efficiency. Finally, I determined percent root colonization by arbuscular mycorrhizal fungi on 3 roots per individual using the ink vinegar staining method and line intercept method under the microscope, highlighting both the presence of hyphae and arbuscules. Let's look at the results. As predicted, the biomass of Paspalum, the dominant species, was strongly reduced under drought, while the biomass of the subordinate species, Cynodon, was not affected. Both species responded to drought by increasing their water use efficiency, estimated by delta 13C in leaf tissue. In this study, we found that the subordinate species Cyanodon was more colonized by mycorrhizal arbuscules than the dominant species, and more interestingly, than root colonization was increasing under drought for the subordinate species, but did not change for Paspalum, the dominant species. Now, I'm going to present you how plant and soil variables were related along the soil moisture gradient that defined the drought stress in our experiment. As shown before, both species, Cyanodont with dashed line and Paspalum in black line, increase their water use efficiency under reduced soil moisture. But we found that the subordinate species Cynodon tended to show higher increase based on the coefficient of the slope. Actually, we found that the increase in water use efficiency for the subordinate species was related to the increased root colonization by mycorrhiza, while no relationship were found for the dominant Paspalum. In our study, we found that soil available nitrogen increased with drought, likely due to a reduced uptake of this nutrient by drought-sensitive dominant species. While the dominant Paspalum maintained a constant concentration of nitrogen in leaves, the subordinate Cyanodon increased its leaf nitrogen under reduced soil moisture. So how is it possible? Well, here again, we found that Cyanodon was able to use a newly available pool of nitrogen under drought through increased root colonization by mycorrhiza. Finally, let's go back to the distinction between homeostatic and flexible species in terms of plant N2P ratio. In this figure, we see that the dominant species Paspalum is clearly homeostatic, as it can maintain a constant N2P ratio independently of changes in N2P ratio in the soil. By contrast, the subordinate species Cyanodon is much more flexible and its N2P ratio in leaf tissue clearly depends on soil resources. During drought, the dominant species is very efficient in maintaining a constant N2P ratio, but looking at its ratio, which is equal to 9, we realize that Paspalum remains limited by nitrogen for its growth. At the opposite, the subordinate species, which is more flexible, was able to increase its uptake of nitrogen and thus increase its leaf N2P ratio, reaching values above 10, which reflects co-limitation in both nitrogen and phosphorus, a better state for plant growth. It won't be surprising to you now that this increase in leaf N2P ratio of the subordinate species Cyanodon, reflecting lower nitrogen limitation, was related to the increased root colonization by mycorrhizal arbuscules. Here is a quick summary of the findings of our study. We got here two strategies: species that are homeostatic, the dominant species, and species that are more flexible, the subordinate species. While stoichiometric homeostasis can be a driver of dominance, it might not be the best strategy to cope with drought. Indeed, reduced soil moisture leads to lower nitrogen mobility in soil, which then can quickly limit nitrogen access to roots, particularly for dominant species which are used to easily uptake nitrogen. If not used by dominant species, the availability of nitrogen can quickly increase in soils, and that's where stoichiometric flexibility and increased mycorrhizal symbiosis can be very beneficial for subordinate species. These species, such as Cyanodon in our study, are capable of continuing taking up nitrogen under drought through the mycorrhizal network, thus increasing their leaf N2P ratio and becoming less nutrient limited. Simultaneously, subordinate species likely benefit from the release of competition from dominant species and also have more access to water provided by mycorrhiza. So what should you keep in mind from our study? Well, first, stoichiometric N2P flexibility allows subordinate species to benefit from drought-induced changes in soil N and P availability. Second, that mycorrhizal plants are more likely to resist drought perturbation due to improved water use efficiency and plant nitrogen uptake under water stress. Third, that facilitated by mycorrhiza, subordinate species can mediate drought effect on plant productivity, something that I already showed in a previous paper. And finally, that diverse communities would be more resistant to drought perturbation, not because of higher number of species per se, but because they contain higher number of subordinate species. Thank you for listening. If you're interested, I invite you to read the full paper which describes more into detail the finding of our study. Or, please contact me if you have any questions.